All right, let's dive into the fascinating world of Enterococcus spp, specifically for our fellow medical microbiologists out there.
Always a hot topic.
Oh, absolutely.
Yeah.
And we'll be using the second edition of Principles and Practice of Clinical Bacteriology by Gillespie and Hawkey to guide us through this deep dive.
Great reference.
It really is. We'll be unpacking the essentials of these intriguing bacteria, what they are, how they cause trouble, and how we fight back. Think of this as your Enterococcus cheat sheet.
Perfect for a quick review or even for those board exams.
Exactly. So, it's fascinating how Enterococcus spp evolved from a misclassification to a significant player in infectious diseases.
Yeah, their journey initially started lumped in with Streptococci.
Right, they were just kind of thrown in there. But something tipped off scientists that these bacteria were different. What was it?
Well, their resilience, for one, and some unique characteristics really set them apart.
Interesting. So walk us through those key features that define Enterococcus spp for our listeners who might need a refresher.
Sure. So we're talking gram-positive Cocci, often found in pairs or short chains.
Okay, so far, so good. Sounds familiar.
But here's the kicker. They're facultative anaerobes.
Meaning?
They can thrive with or without oxygen.
So they're not picky.
Not at all. And they also have this interesting quirk sometimes showing a weak pseudo-catalase reaction.
Ah, those pseudocatalyses. Always a fun surprise in the lab.
Right. But that's not all. These guys are tough. They thrive in conditions that would send other bacteria running for cover.
Really? Like what? Give us an example.
Imagine a petri dish with 6.5% sodium chloride.
Okay, pretty salty.
Most bacteria wouldn't stand a chance, but Enterococci, they're multiplying like crazy.
Wow, so if it can handle salt like a champ, it's probably Enterococcus.
Exactly. And they're not just salt tolerant, they can handle heat too, growing comfortably at temperatures up to 45 degrees Celsius.
That's pretty impressive.
Plus, they have a thing for bile.
A thing for bile.
They can hydrolyze esculin in the presence of bile, something other similar bacteria struggle with.
So salt, heat, bile, and Enterococci are like the bacterial version of an extreme athlete.
I like that analogy.
And speaking of extreme environments, where can we find these resilient microbes? I know they're in the gut, but anywhere else surprising?
You might be surprised to learn they're more widespread than you think. While the GI tracts of humans and animals are their primary home, we've also found them in insects, plants, and even some fermented foods like cheese.
Cheese? Really?
Oh yeah, they're masters of adaptation,
Yeah.
which is probably why they do so well in places like hospitals.
That makes sense. But here's the million-dollar question. How did these seemingly harmless gut dwellers become opportunistic pathogens?
Well, even before we were using antibiotics so widely, Enterococci were known to cause infections, particularly endocarditis.
Serious heart condition.
Exactly. And also those tricky infections in the pelvic, intra-abdominal, and urinary areas.
Okay, so they're not so harmless after all.
Nope. They have this whole arsenal of virulence factors that allow them to not just survive, but to thrive and cause some real havoc.
So what kind of weaponry are we talking about here?
Their virulence factors fall into three main categories: secreted factors, surface-located proteins, and then those cell wall polysaccharides. Each plays a unique role in their ability to cause infection and outsmart our immune system.
A three-pronged attack. Let's start with those secreted factors. What do they do?
Well, one of their most potent weapons is cytolysin. Think of it like a demolition crew, breaking down red blood cells.
Cytolysin, the bacterial wrecking ball.
Exactly. And that's often why we see hemolytic activity with certain Enterococcus strains.
Okay, I'm starting to see how they cause so much trouble. Anything else we should be wary of?
Oh yeah, Enterococcus also uses this duo, gelatinase and serine protease.
What do those do?
Think of them as molecular scissors. They snip away at various proteins, even collagen.
Collagen. That's the scaffolding that holds our tissues together.
Exactly.
So they're not content with attacking cells directly. They're dismantling the infrastructure too.
Right. And this protein-degrading ability likely fuels the progression of the infection. But it doesn't stop there.
Oh no.
They also have surface-located proteins that act like grappling hooks, allowing them to latch onto host cells.
Okay, so let's talk about these surface-located proteins. What are some of the key players?
One that comes to mind is aggregation substance. This protein helps them stick together, but it also has other functions.
Like what?
Well, it allows them to bind to human cells and, believe it or not, even sneak into host cells undetected.
Talk about stealthy. Are there any other sneaky proteins we should know about?
Well, there's ESP, short for enterococcal surface protein. It's basically their master key.
A master key.
Yeah, it lets them colonize various sites, particularly the urinary tract. It also helps them build those notorious biofilms.
Biofilms. The bacterial fortresses that make treatment such a headache.
Exactly. And finally, there are those cell wall polysaccharides.
What role do those play?
These complex sugars act as a disguise, shielding Enterococcus from our immune system.
Clever camouflage. Any particular polysaccharide we should be aware of?
EPA, or enterococcal polysaccharide antigen, is a big one. It's linked to increased virulence, biofilm formation, and resistance to those all-important phagocytes.
So EPA is like their invisibility cloak, allowing them to slip past our defenses. It's amazing how these seemingly simple bacteria have evolved such a complex arsenal.
And here's something interesting. Most of the research on these virulence factors has focused on E. faecalis, the species we see most often in the lab.
Yeah, E. faecalis, the usual suspect.
Right. But when it comes to E. faecium, the rising star of nosocomial infections, well, we still have a lot to learn about its virulence mechanisms.
E. faecium, the up-and-comer with a penchant for hospital settings. It sounds like we have a lot more to uncover about this one. So last time we were talking about E. faecium and how it seems to be, more and more common in hospitals.
Right, those nosocomial infections.
Yeah, exactly. We were also talking about how it seems like we just don't know as much about it.
We do know it's become a real challenge in healthcare settings.
And I know we touched on it briefly, but I think it's worth revisiting. VRE, vancomycin-resistant enterococcus. It's a pretty intimidating acronym.
It is, and for good reason.
Yeah. Can you remind us why it's such a concern, especially in those hospital settings we were just talking about?
Well, a key factor is the what we call the colonization pressure.
Colonization pressure? What's that?
It's basically the prevalence of VRE colonized patients in a specific setting, like a hospital. So the more patients carrying VRE, the higher the risk of it spreading.
So more VRE equals more chances of transmission and new infections.
Exactly. And it's not just about, direct patient-to-patient spread.
Oh, right. The environment itself can become a reservoir for VRE.
Unfortunately, yes. These bacteria are pretty hardy. They can survive on surfaces for quite a while, and that's where things like thorough disinfection come in. It's absolutely critical.
Oh, absolutely. Thinking about all those high-touch surfaces, bed rails, door handles, all the medical equipment, everything.
All potential hiding spots for those tenacious microbes. So hand hygiene, proper use of gloves and gowns, not sharing equipment between patients, those are all essential practices for healthcare workers, really everyone in the hospital.
It's a team effort, for sure. No weak links in the chain. Now, moving on to the clinical side of things, we talked about the general principles of managing enterococcal infections. But what about the specifics? Like, how do we approach different types of infections? Can you give us some examples?
Sure. Let's say we have a patient with a uncomplicated urinary tract infection, a UTI.
Okay, a UTI caused by Enterococcus. What's the game plan?
Well, as we discussed, for uncomplicated infections, often a single antibiotic can do the trick. Ampicillin or penicillin are usually good choices.
Unless, of course, the patient has a beta-lactam allergy.
Right. Then vancomycin would be the alternative. And specifically for UTIs, nitrofurantoin and phosphomycin can also be good options.
Gotcha. Good to have those in our arsenal. But what about when we encounter those multi-drug resistant strains, like VRE? Does that change things?
Well, if it's a VRE strain, but it's still susceptible to ampicillin, we might still be in luck.
Okay, so ampicillin can still be effective.
Potentially, yeah. Even at higher doses, it can work in many cases.
Good to know. But what about those VRE strains that are resistant to ampicillin? What are our options then?
Well, for VRE UTIs, especially if they're limited to the lower urinary tract, nitrofurantoin or phosphomycin might still be viable options. But for more serious VRE infections, we might need to consider other antibiotics.
Like?
Well, quinipristin delphapristin is one option.
And quinipristin delphapristin, that's a mouthful. Is that marketed as Sinercid?
That's right, Sinercid. It's a combination of 2 streptogrammin antibiotics.
Okay, and it's effective against VRE.
Yes, it can be quite effective, especially against E. faecium. It works by inhibiting bacterial protein synthesis.
So it basically stops the bacteria from making the proteins they need to survive.
Exactly. But it's not without its downsides.
What kind of downsides?
Side effects can be an issue for some patients, and it has to be administered intravenously.
Okay, so that can be inconvenient for some patients. What about linezolid? I remember we talked about that one as well.
Linezolid is another option for treating VRE. It also works by inhibiting protein synthesis. but at a different step than Synarcid.
So it's effective against some strains that might be resistant to Synarcid.
Exactly.
But I think you mentioned earlier that there are already some concerns about resistance to linezolid.
Unfortunately, yes. We have seen some cases of linezolid-resistant Enterococcus.
So even with newer antibiotics, resistance is a constant concern.
It really is. It highlights the need for responsible antibiotic use and for the development of new therapies, of course.
Absolutely. Now, I want to talk about endocarditis. It's a much more serious infection.
It is. It requires a much more aggressive approach to treatment.
And typically that involves a combination of antibiotics.
Right. Usually penicillin or ampicillin along with an aminoglycoside, like gentamicin or streptomycin. That combination is really important for getting those bactericidal effects, actually killing the bacteria.
But what happens when we have that high-level aminoglycoside resistance? You know, those cases where our usual strategy is off the table.
Well, for epithecium, high-dose ampicillin alone might be effective in some cases, especially if the minimum inhibitory concentration, the MIC, is low.
So we're basically trying to compensate for the lack of that aminoglycoside synergy.
Right. And for E. faecalis endocarditis, ampicillin is usually a good choice, even if the strain is vancomycin resistant.
Okay, so ampicillin still has a role to play there. But what about those really resistant strains? The ones that are resistant to both ampicillin and aminoglycosides? Do we have any options left?
Those cases can be really challenging. We might have to consider some less conventional antibiotics, like daptomycin.
Daptomycin. Remind me, how does that one work?
It's a lipopeptide antibiotic. It targets the bacterial cell membrane.
Okay, so it disrupts the membrane, causing the bacteria to essentially leak out their contents.
Right. And it's shown some promise against multi-drug-resistant Enterococcus, including VRE.
So it's another weapon in our arsenal. But I imagine, like all antibiotics, we need to be careful about using it judiciously. We don't want to fuel more resistance.
Exactly. Another option for those resistant cases could be tigecycline.
Tigecycline, that's the glycylcycline antibiotic, right?
That's the one. It inhibits protein synthesis, and it has shown activity against some VRE strains. But it's important to note that it's not FDA approved specifically for enterococcal infections.
So we're using it off-label. It's like we're in a constant game of chess trying to outmaneuver these bacteria.
Exactly. And sometimes, despite our best efforts, we have to consider other options, like surgery.
Surgery for Enterococcus infections.
Well, in some cases, yes, especially if the infection is causing serious damage or if it's just not responding to antibiotics.
Okay, so we're talking about those really severe cases, like endocarditis, where the infection is affecting the heart valves.
Right. In those cases, surgery might be necessary to remove infected tissue or even replace a damaged heart valve.
So it's a multidisciplinary effort. We need the expertise of infectious disease specialists, cardiologists, maybe even surgeons.
Absolutely. A collaborative approach is essential for these complex cases.
Now, I want to touch on something we mentioned earlier, personalized medicine. How does that fit into the management of Enterococcus infections?
Well, it's all about tailoring treatment to the individual patient.
Right, not just using a one-size-fits-all approach.
Exactly. We need to consider the patient's entire clinical picture, their age, any underlying health conditions, their medication allergies, any previous antibiotic exposure, even their genetic makeup.
So we're gathering as much information as possible to make the most informed decisions.
Yes. The goal is to choose the right antibiotic at the right dose for the right duration, all while minimizing the risk of side effects and preventing the emergence of resistance.
It's like creating a custom-tailored treatment plan for each patient.
You could say that. And as we learn more about how host factors, bacterial virulence, and antibiotic resistance all interact, we can get even better at this personalized approach.
It's really the future of medicine, isn't it?
It is.
Now, speaking of the future, what's on the horizon in the world of Enterococcus research? What are scientists excited about?
Well, one area that's really promising is the development of new antibiotics, ones that target Enterococcus in completely different ways.
So not just relying on the same old targets.
Right. Researchers are exploring antibiotics that disrupt the bacteria's virulence mechanisms or interfere with their ability to form biofilms.
So not necessarily killing the bacteria outright, but making them less harmful?
Exactly. The idea is to disarm them, so to speak, make them more vulnerable to our immune system or to other antibiotics.
That's fascinating. What about vaccines? Any progress there?
There are actually several vaccine candidates in development. Some are targeting specific proteins on the surface of Enterococcus.
What are those proteins doing?
They're involved in adhesion and colonization.
So if we can block those proteins, we can prevent the bacteria from sticking around in the first place.
That's the goal.
Pretty ingenious. Are there any other novel approaches being explored?
There's a lot of interest in phage therapy.
Phage therapy. Remind me what that is again.
It involves using bacteriophages, which are viruses that specifically infect and kill bacteria.
Oh, right. I remember now. So we're basically pitting viruses against bacteria, letting them fight it out.
In a way, yes. And another area of research is focusing on the gut microbiome.
The gut microbiome. That's become such a hot topic in recent years.
It has, and for good reason. We're learning more and more about how the gut microbiome impacts our overall health, including our susceptibility to infections.
So how does this relate to Enterococcus?
Well, scientists are looking at ways to manipulate the gut microbiome to make it less hospitable to Enterococcus.
So it's like we're trying to create a more hostile environment for the bad guys.
Right. We can try to boost the populations of beneficial bacteria that can compete with Enterococcus.
It's fascinating how much we're learning about the gut microbiome and how it influences so many aspects of our health.
It really is. And as we learn more, we're likely to find even more innovative ways to prevent and treat Enterococcus infections.
It sounds like the future of Enterococcus research is bright.
It is. It's a constant challenge, but we're making progress.
All right, welcome back to our Enterococcus deep dive.
The final round.
That's right. It's time to put that knowledge to the test.
Quiz time.
Exactly. So over the past 2 episodes, we've journeyed through the world of Enterococcus, exploring, their unique characteristics, their virulence factors, the clinical challenges they present.
We've covered a lot of ground.
We have. And now it's time to solidify that understanding with some, challenging exam-style questions.
Think of it as a final exam.
Perfect way to put it. So are you ready to dive in?
Let's do it.
All right. First question. Which biochemical test reliably distinguishes Enterococci from most streptococci? It's a classic microbiology question.
That would be the PYR test. And Cocci are generally PYR positive.
And streptococci.
PYR negative?
Yeah,
Usually.
Exactly. The PYR test, our trusty tool in the microbiology lab. All right, next question. What is the primary mechanism of vancomycin resistance in Vanna-type Enterococcus? This one really gets to the heart of the VRE challenge.
Okay, so Vanna-type Enterococci, they modify their peptidoglycan precursors. Specifically, they replace the usual D-alanine D-alanine ending with D-alanine D-lactate.
Right, a subtle change with big consequences.
Huge. This alteration significantly reduces vancomycin's ability to bind, which basically renders the drug ineffective.
It's like they've outsmarted our best defense.
They're always one step ahead.
All right, moving on. Which 2 antibiotics are currently FDA approved for treating VRE infections? This is crucial for anyone dealing with VRE in the clinical setting.
We've got quinupristin, dalfapristin, that's Sinercid, and then there's linezolid.
2 important weapons in our fight against those resistant strains.
Absolutely.
Okay. Now for a question about those tricky aminoglycosides. Why is high-level gentamicin resistance, HLGR, a concern when treating enterococcal infections? This one requires a bit more thinking about how different antibiotics work together.
Well, the big concern with HLGR is that it disrupts the synergy we normally rely on. When we combine aminoglycosides with cell wall active agents like penicillin or vancomycin.
So we lose that 1-2 punch.
Exactly. And that synergy is really important, especially for serious infections like endocarditis. We need that bactericidal effect.
It really limits our options.
It does.
All right, final question. What is the significance of the colonization pressure in the spread of VRE? This one goes beyond the individual patient and looks at the bigger picture of infection control.
Colonization pressure. So that refers to the prevalence of VRE colonized patients in a particular setting, like a hospital, for example. And a higher colonization pressure means a greater chance of transmission and more new infections.
So the more VRE circulating, the harder it is to contain.
Exactly. It underscores how important infection control measures are. Hand hygiene, environmental disinfection, being mindful of antibiotic use.
Absolutely. It's a team effort to keep these resistant organisms in check.
Couldn't agree more.
Well, it seems like you aced those exam questions. Congratulations.
Thanks. I've been studying.
Clearly. And for our listeners, we hope this deep dive into the world of Enterococcus has been, informative and engaging.
They're fascinating organisms.
They are. And they present unique challenges in the clinical setting. But the world of microbiology is always evolving.
New discoveries all the time.
Exactly. So stay curious, keep learning, and never underestimate those tiny but mighty microbes.
They can really pack a punch.
That's for sure. And remember, the fight against antibiotic resistance is an ongoing one.
We all have a role to play.
Absolutely. Judicious antibiotic use, robust infection control measures, those are key.
Key to keeping those life-saving medications effective.
Well said. So until next time, keep those microscopes focused, those minds engaged.
And may your future encounters with Enterococcus be both enlightening and successful.
Couldn't have said it better myself. Thanks for joining us on this deep dive into the world of Enterococcus. We'll see you next time for another fascinating exploration into the world of microbiology.